Hello Year 12, welcome to another lesson. You'll be happy to notice it's not statistics anymore. We're going to a different topic. We're going to look at genetic diversity. So to start off with a little bit of knowledge retrieval, so what I'd like you to do is think about the next question. So I think you should be into a bit of a routine by now. So here's a particular question I'd like you to have a look at and think about. Okay, so explain the difference between substitution, deletion and addition mutations and the effects of such mutations. So if you just pause the screen now, jot down your ideas, your thoughts, and then we'll pick things up in a moment. So pause the screen now. Okay, hopefully you've gone through that and you can recall those particular mutations from a few weeks ago. So if we're looking at the first one, we're looking at the idea of substitution. That's quite simply, the name suggests it, we're substituting or changing one base for another. So the example I've given you there, ACT, GGC, ATA, and that first thymine we're substituting in guanine. So a slight change in the actual sequence. The next one, deletion. Again, as the name suggests, we're removing a base or a number of bases from the sequence. So in this case, I start off with ACT, GGC, ATA, and I've removed one of the guanine bases from that particular sequence. So that's a deletion. And addition, again, as the name suggests, fairly straightforward. We're including, adding an extra base or bases into the sequence. So I've got ACT, GGC, ATA, and I've added an extra thymine into that particular sequence. Consequence of these mutations would vary. So the simplest would be quite simply there's no change. So because the genetic code is degenerate, we may still code for exactly the same amino acid. So even though there's been a mutation or a change in the base sequence, it hasn't had an impact on the genetic, on the actual amino acid sequence if we're producing protein. It could be that we're coding for a completely different amino acid or a number of amino acids. It may be that we're introducing punctuation. It could be a stop codon, for example, that we're now coding for because of the particular mutation that we've looked at. Or we could get something like a reading frame shift where the sequence that we've got has moved along slightly one way or another, and that could be a number of different amino acids that are now coded for and totally change the protein, if indeed this was a particular sequence that led to production of a protein. So that's a bit of a recap on some of these things to do with mutations. So we're going to move on and look more in detail about what we mean by genetic diversity. So just some terminology just to recap before we begin. The first one, really important one, is just this term an allele. This is back from GCSE, so it shouldn't be too much of a challenge. And if I was in lesson, I'd be quizzing one or two of you right now saying, what's an allele? I'm going to think of an allele quite simply. It's a different version of a gene. So I've got two pictures here at the right of the screen looking at chromosomes from mother and father. We can see we've got different alleles on the chromosome from mum compared to the allele on the chromosome from dad. So even though it's a gene for a particular characteristic, it has a different version. The term genetic variation, it's important we've got an idea of a good definition for this. The definition that we really need to be confident with is the number of different alleles of genes in a population or a species. I'm not a huge fan of the term genetic variation. Allele variation I think would be more useful. We're looking at genetic variation, the number of different alleles of genes in a population or in a particular species. Okay, so it is a definition that's worth committing to memory. Therefore, if we've got particularly high genetic variation within a population, we are looking at a high number of different alleles within that population. And the consequences of this are really quite important. So in terms of genetic variation, if we have that high level of variation, that greater variety of different alleles within the population, that's going to give a particular population, a particular species, a greater likelihood for survival, a better chance of survival. They're more likely going to be able to withstand any environmental change and evolve as a population as time goes on. Wider range of alleles leads to a wider range of characteristics, and therefore there's a higher probability that any individual within that particular population would possess the appropriate characteristics to survive in that new environment as the environmental change develops. So really important that populations do have genetic variation to withstand that impact of environmental change. So where does the variation come from? Go back and think about some principles we've already addressed at the start of this particular YouTube video clip. And in previous lessons, we've got things like meiosis, or the idea of independent segregation and crossing over can bring about increased variation within the offspring. That's one of the benefits of having sexual reproduction, that idea of variation being introduced. And we've also looked at mutations and the consequences of those. Both of these particular events are huge contributors towards enhancing and developing genetic variation. But there are things that occur within populations that can reduce genetic variation. So we need to kind of take those into account too. So in terms of reducing genetic variation, this is going to reduce the number of different alleles within a population. If you're reducing the number of different alleles, you're reducing that variation, it's more likely that a particular population will be less successful at surviving a period of environmental change. So it's a really important principle that if we are doing things that reduce genetic diversity, or things occur that reduce genetic diversity, it can have quite adverse effects on a particular population and a particular species, because we're reducing the number of different alleles that are there. Now, when I do this particular topic, I like to use an example in my own garden. So the next clip is my pond. I've had this pond for, I think about 8-9 years now. So I built it and put it in several years ago in the garden. And if you've gone back 6 to 8 weeks and had a good look in my pond, what you would have seen in my pond is about 40 to 50 frogs. So you can't see them at the minute, they've kind of moved away currently, but there would have been 40 to 50 frogs in there breeding. And me being the biologist that I am, I'd often come out and have a look on a night and come up with a torch and just see how many frogs I could find. I want to notice there's a huge variety in the skin colour of the frogs in the pond. Some frogs I see every year. I can recognise some of them. They've got a particular sort of skin pattern that's kind of identifiable year on year, but a huge population. This now pond, you can't see them in that clip, but it's actually full of tadpoles. We get toads visiting as well. But in my particular population of frogs, I said there is a variety of different skin colours. And that's come about because of different allele combinations and genetic variation and diversity within the frog population in my particular garden, in my particular area. So if we're going to think about what leads to that, the skin colour within frogs, it would be a number of different genes involved in giving the particular skin pigments and patterns within the skin, and a number of different alleles for those genes as well. So if we represent those genes and alleles as beads, okay, so the next clip, I've got a bottle here with a number of different coloured beads, and if each bead in there represents, and just using the characteristic of skin colour for a moment, just to keep it quite simple. If those different beads in there represent the variety of different alleles responsible for the particular skin colours and pigments on my frog population, what can happen under some circumstances, there can be an event that results in a drastic reduction in the population size. And this particular event is known as a genetic bottleneck. So here's my original sample size. Okay, I've got a number of beads in there, and therefore there's quite a large population of frogs responsible. I said 40 to 50 in my pond. Something might happen that reduces the population size. So from 1 year to the next, all of a sudden I'm down to a tiny number of frogs compared to what I originally had. Consequence of that is a much smaller variety of alleles compared to the original population. Okay, so if that new population of frogs that has a much more reduced variety of alleles for skin colour and pigment and so on, if they're then going to start and form a new population, that surviving population has that reduced variety, and therefore the descendants of that population would also have that reduced variety. So in this clip on the screen, you can see the original population had blue, white and yellow, but the bottlenecking event, that dramatic reduction in the number of surviving organisms and therefore the number of available alleles within subsequent generations has decreased. So that new population, it's going to breed and reproduce and produce further offspring, but the variety of alleles within that surviving population is significantly different compared to the original population. So that's what we call a genetic bottleneck. Okay, and that's often as a result of a dramatic reduction in the population size. We'll have a look at some examples of that in a few minutes' time. Another event that's also really well known is an event known as the founder effect. So the founder effect is where a population forms a new kind of offshoot, a new population elsewhere. So the example in this little picture here shows a particular island at the left, where we've got a variety of different coloured ladybirds probably in this example, so it's some kind of beetle. We've got red and yellow within the original population, and through some sort of natural event, whether that's a storm or some sort of strong winds or whatever it might be, a small proportion of that original population are moved and they form a new population elsewhere. So they're founding a new population. In this case, the idea is on an island, so it's away from the original population, and that new population that's on that island would then reproduce and colonize that particular island and form a new population. Again, because the new individuals on that island have reduced variety of their alleles, reduced genetic diversity, because not very many of them moved to the island. That new population also has reduced genetic diversity. So you can kind of see these two events, this genetic bottlenecking and the founder effect, both have similar outcomes. We've got a reduction in a population size, a new, smaller population that will interbreed and subsequently colonize that new environment, but as a consequence there's a reduction in genetic diversity, and that can have significant impact for the organisms involved. So I said we'll look at a couple of examples. So there's 4 that I want you to go away and just kind of look into, just to kind of cement the ideas about genetic bottlenecks and the founder effect. So the 4 I'd like to have a look at are quite well known as examples of each of those principles. So the top 2 involve the cheetah and the northern elephant seal, and the bottom 2 are human examples involving a condition called Huntington's disease, and the other one looking at Ellis-Van-Creveld syndrome in the Amish in North America. I'm going to give you, in the slide that I send out to you, and so the handouts that I send out to you, I'm going to give you a number of things I'd like you to do. These are the websites that are particularly useful for assessing each one of those particular areas. So if you're going to be looking at Huntington's disease, there's a number of questions I'd like you to try and answer. It's a good example of the founder effect. So Huntington's is, why is the presence of Huntington's disease in certain South African populations an example of the founder effect? So find out a little bit about what it is and why does that kind of meet the criteria for the founder effect. How has it occurred? What are the implications for those particular populations? And can anything be done? Sometimes something can be done and sometimes it can't. So what kind of, is there anything that can be done to address those particular conditions in that population? Number of websites to have a look at, and there's one all about cheetahs, there's one about the elephant seal, there's a couple to do with Huntington's, and there's one to do with the Ellis-Van-Creveld syndrome of the Amish. This is the kind of thing you can fill in, so it's just a brief summary of what is the founder effect and then examples for each, Huntington's and Ellis-Van-Creveld. And for genetic bottlenecks, have a look at the northern elephant seal and the African cheetah. Any problems, as usual, drop me an email, and what I'd like to see you sending back to me is a photograph or a clip of just that slide at the end. So you could take your own slide, do as a PowerPoint slide, email that back to me, take a photograph of your work, email it back, to just tell me a little bit about those 4 particular examples of 2 for the founder effect and 2 for genetic bottlenecks. Okay, good luck year 12. Hopefully speak to you soon. Any problems, as usual, drop me an email and I'll speak to you in the near future. Bye for now.